Okay, let's get started. Okay, as always, good morning to American colleagues, good afternoon to the European colleagues, and good evening to our Asian colleagues. Today, we are very happy to invite Professor Christophe Rabouille from France and University of Paris to give a talk about organic carbon from the Congo River. And so this is a very interesting river, and because also not only one of the largest river, but also with the canyon and the discharge and transport directly to the deep sea fan. So before I introduce Christophe, and I want to see a little bit like what I said this Wednesday. So far we have already 50 webinars already archived on our YouTube Source to Think YouTube channel. But as I said last time, from beginning, we try to be all inclusive, try to be diverse as much as possible. And it's not easy, but we're still working on that. So if you have any colleague, any even your student or postdoc, if they also can contribute Source to Think related talk, and please recommend it to me. Even as I always welcome anybody if you want to volunteer a talk, and particularly for our women colleague, people of color, underrepresented group of people, and people from developing country, no matter you from Vietnam, Nigeria, or even Chile. So if you are a crazy lover of the river and the delta, we will warmly welcome you to contribute a talk. This will make this community and Source to Think much better, because this is the global issue. We talk about global river and the delta system. We cannot only invite American professors. No, we cannot do that. And you involvement, you participate is very important for us. And even your English is not the first language like me. My English is not good. My kids always make fun of me, my accent. But as soon as you have a passion, this is my student love me very much. As soon as you love the river and the delta, you have the passion to do this, you can do it. So we're very open. And please, particularly for those rivers we haven't covered yet, like the Russia River, I tried very hard, to invite some Russia colleagues, and not really successful yet. So if you know somebody, I send an email, but sometimes no reply. But if you personally know them, contact them, copy me, and so we make better. Okay, so I think said enough. And so before I introduce Christophe, next week, next Wednesday, Hugh Sinclair, he's also here. He will talk about, he's from University of Edinburgh. He will talking about the Himalaya, the Ganges system. It's great. And today I'm moving a new house at Edinburgh Drive. That's great. And okay, Christophe graduated from University of Paris in 1990. And is a geochemistry, I think is very close to what the Bob Allen's doing. And also a postdoc at University of Brazil and sabbatical, University of Hawaii, and also visiting professor in the Xiamen University. And current research scientist at the lab of the Science Environment Center. So his research mainly is, the coastal biogeochemical chemistry, sediment diagenesis, and the Institute of Observation Carbon. And so he worked on so many large river systems, including the Congo, in the Gulf of Lyon, and other systems, even the Pearl River in China. So I think, Christophe, are you ready? So now you can share your screen for the presentation mode. We're looking forward to your talk.
Okay. 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 Thank you, Paul, for the. Okay, can I?
Yes, yes, okay. It's good.
So thank you, Paul, for the nice introduction. Today I will talk about one of my favorite research project, which was on the Congo River, and especially its deep sea fan that we investigated almost 10 years ago. And we worked on it for the last decade, and we still published a paper last year, I guess. And so today my talk will be about the routing of organic carbon from the river to its deep sea fan. I will try to let you understand how this system works and what the peculiarities are. I have two main co-authors for this talk, which is the first one, François Baudin from the ISTEP at the Sorbonne University in Paris, and the other one, Bernard Denielou, who is at Ifremer in Brest, France. And I like also to acknowledge my collaboration in the lab, which are on the below list here. Oh, I will take that. Okay, here Bruno Bomblet, Cécile Catalot, Claire Trainier, Flora Toussaint, Jean-Louis Reis, Nadine Tisnera, and Laura Pozzato. So, okay, let me do something for that. Okay, right. Okay, so Paul asked me before I go into the main subject to make some advertisement for other work in case there are some PhD students or postdocs that don't know much about my work. So just very quickly, I would like to let you know that we've been working for almost 20 years on the Rhone Delta in the Med Sea. So we have several papers on that topic. Most of them dealing with sediment, recycling, geochemistry, burial, carbon source, and so on. We have tried to make an understanding of the extreme events, so these extreme floods, on sediment deposition, organic carbon processing, storage, and also on the alkalinity and DIC exchange between the sediment and the water column. We had these two papers by Rassman in 2016 and 2020. Recently, we had some work on the Mississippi Delta and the Louisiana Shelf. So this is just a new one, which is out in Frontiers just now. And another one by Owings et al. On the shelf and canyon slope side. So these two papers, one in marine chemistry and the other one in Frontiers in marine science. And I also have worked a lot on coastal lagoons, on the human influence on these lagoons. In one of the French lagoons of the Mediterranean, which is called the Thau Lagoon. This was like more than 10 years ago. And more recently on the Venice Lagoon with Daniele Brugolin from Venice. And that's also in Frontiers in 2021. I've also worked on the Pearl River with Minhan Dai's group. This is still in prep, although this was done some years ago already. Okay, now let's go to the main topic of my talk, which is on the Congo Canyon and deep sea fan. The introduction just features the fact that the ocean margins are obviously constrained by the fluxes at their boundaries. There are small, relatively small bodies of water compared to the ocean or to the land nearby. And of course, this exchanges with these two boundaries constrain our carbon budget. Of course, there is this input from the rivers that we will deal a lot with today, but there is also the input from the ocean, onwelling, production of marine carbon. And so these two forcings play a very prominent role in the biogeochemistry of the ocean margins. Of course, the ocean margins also export particulate matter. That's what I want to focus on today, to the open ocean. There are several different pathways. The one that I will investigate today on the West African margin is the canyon channel transport. And you will see that it's quite an important transport in that area. So for those of you who attended the talk by Peter Tolling last year, I guess, is talked about the export processes by turbidity current and the role they can play in exporting sediments from the shelf or the river estuary to the deep sea. And he showed that the processes that deal with this export, they are really under study. So there's very few studies that have started like maybe 10 years ago, maybe 5 years ago. And it's not very well known, but he really insisted in the fact that these turbidity currents play a major role in exporting the material close to these very large canyons. So of course, because of the lack of knowledge of these processes, the mass balance for carbon in these regions is not well established. So that's why we went to this zone and tried to establish a mass balance for organic carbon. And so in this talk, I will try to shed light on these questions of the organic carbon budget in the context of the Congo Canyon. Remember that the Congo is the second largest river on Earth. I will just emphasize that now. So this characteristic of the Congo River has a discharge of 40,000 cubic meters per second. It's the second largest river on Earth. Because of it's a rainforest, which is on the equator, it has a quite stable water flow from 30,000 to 60,000 cubic meters per second. And the particulate discharge for this water flux is relatively small compared to the Amazon, but it's still very large compared to most of the other systems. And the thing is that I didn't put that together as Paul Tolling did last time, but it has a giant underwater canyon, and this canyon is that giant because it hasn't moved for several millions of years probably. It's expanding more than 800 kilometers offshore, so it's a very large system. And you can see that on this slide, the river mouth is somewhere here, and here it's 800 kilometers further out at sea. Is this terminal lobe complex. And this extends from nearly 0 meter depth or 100 meter depths in the estuary to almost 5,000 meter in the abyssal plain. So it's really a projection of the river and the estuary out at sea. Remember that 1,000 kilometers away from the shore and 5 kilometers below the water surface. If we go more into the details and we look a little bit more how this system works, it is very special because the canyon head is in the estuary, it's somewhere here. So there is an incision in the shelf, like 150 meters that goes right into the estuary. And so there's a direct connection between the river and the canyon. So there's accumulation of particulate material. Paul Tolling described that very well. Near the shelf break, and we don't know exactly, probably by a combination of tides and floods, that there are some turbidity currents. The turbidity activity has been reported for very long by breakage of submarine cables and also recent turbidites in the Holocene that were recorded in the sediments. And also we had also moorings that were broken by this turbiditic activity. And what I will be talking about today is the fan, so the lobe zone here, which is a terminal lobe complex where the material is ultimately deposited, and it is fueled by turbidity currents, of course. If we look at the cross section of this canyon, one feature of it is that it is the incision of the canyon in the slope is at the apex near the shelf break is 1,400 meters. I went to the web and I looked at the Grand Canyon. For the American that knows this particular place, it's the same side. So if you were on the shelf looking at the canyon itself at this particular place here, you would look like if you were looking down the Grand Canyon. So it's really impressive. It's a very impressive canyon depth here. And then the profile slows down, and then here to the lobe entrance, where I will focus my my talk afterwards. So we were lucky enough to have 2 cruises in 2011 for a total of 40 days at sea on the Pourquoi Pas, this big ship of the Ifremer, and it's a ROV carrying vessel. So the ROV that we use here was the Victor 6000, and it's a very useful and powerful tool when looking at this depth. Remember that this is 5,000 meter depth. We use high technologies, high resolution mapping and imagery by the ROV. Also, we use the ROV operated microprofiler benthic chambers, and also autonomous benthic chambers on microprofilers. And we use a lot of gears associated with the ROV, cores, box cores, nets, water, etc. So we use everything we could with this ROV stuff. We did like 15 dives of 1 to 2 days. So this was really a ROV-based cruise. And we also backed up all these techniques by classical techniques, multi-coring, piston coring. You will see that it's very important in terms of measuring the carbon balance. Okay, so the general strategy for studying these lobes, so this is the small area that is at the end of the channel levee systems, so was to look at a spatial gradient from the lobe entrance to the lobe main deposition. So this site is the actual site of turbidite deposition in the complex. And we also looked, I will not go into that today, but maybe another time. We also looked at the temporal gradient, because there are other sites in the north which are close, which used to be fed by a previous channel here, and so this is an old site that's been abandoned by recent turbidites, probably for 4,000 to 5,000 years, and so it gives us the opportunity to look how a site evolves afterwards. But I will not go into that. If you want to know more about the geology, morphology, structure, composition, build-up processes of these lobes, you can refer to this paper by Bernard Degnelou et al. In the Deep Sea Research special issue that we produced at the end of this Congo project, and it's the volume is 142. So that was in 2017. So if you want to know more, I will refer to this paper during my talk. But if you want to know more, go to this special issue, and all the details and everything, the interesting bits for you will be there. I will not leave you without talking of my favorite tool, which is the Sediment Autonomous Profiler. So this one was positioned by the ROV. It's based on the use of microelectrode that penetrates the sediment. We use 5 oxygen, 1 resistivity, and some pH profiles also. We did some spatial resolution of 0.2 millimeters, so that allowed us to have very precise and in situ measured oxygen profiles. Here is a view of oxygen electrodes working in the sediment. We also used this profiler on an autonomous vehicle coupled to benthic chambers that allowed us to have the total oxygen uptake of this zone. And this is the Ifremer RAP 2 that you'll see here, manipulated on board the Atalante, I guess. And so this allowed us to have a local coupling between the diffusive and the total fluxes measured in situ. We also deployed sediment traps here to estimate the vertical flux of organic matter to the site. And as I said before, we also use sediment coring by multi-coring here because the sediment is so soft. Look, we had to adapt some larger fits so that the the multi-core will not sink into the mud. The mud is very unconsolidated. And we also did some piston coring, 15 to 20 meters, to assess what's going on below the surface sediments. Okay, now for the carbon budgeting methods, because of course in that area, due to turbidity currents, most of the flux, or a good share of the flux, we believe, was carried by the lateral flux. We said, okay, let's try to get the input flux by a sum of the burial flux on the recycling flux. Of course, this somehow assumes that there is a steady state, long-term or short-term steady state. So we can discuss that in the discussion afterwards if you want. But this is a way that we found to recalculate the input flux to these sediments and to the lobe in general. So what we did is estimate recycling by the oxygen consumption, and we calculated the POC recycling by the O2 consumption multiplied by a factor that I will tell you more about, and we also calculated and measured the burial flux so that we sum to get the input flux to the lobe for the carbon. Yeah. So, but before I go into this this carbon budgeting, which you are all expecting, I suppose, I would like to just say a word on the peculiar ecosystems, because this was really a shock and a surprise when we went out there. So these are organic rich sediments, and during these two expeditions with the ROV surveys, we discovered and described some dense habitats that you can see. One example here, made of reduced sediments, symbiotic bivalves. You will see that that are known from the slope coal sieves and bacterial mats. So before I discuss the carbon budgeting, I will go on and tell you a bit more about these ecosystems. First, what we did, because we had the chance to have the ROV survey of the sediment, both for the precise mapping of the bathymetry of the zone, and also with a closer look at 8 meters with photographs on the transects, we were able to map the different habitats that we have there. And mostly what we found was a succession of discrete reduced sediments, which on this graph are the black dots here, and with sometimes some bivalves associated with that. This is the red dots here, and for each different station, we were able to map these dense habitats, and so the density mostly increases towards the most recent lobe, which is called Site C, and it contains a large diversity of habitats, as you can see here on this map. I will not go into the details. If you want to have more information about that, you can refer to this Sen et al. Paper in the deep sea special issue. Now I have some movies that Paul will run. Paul, can you run the movie? Oh, thanks. So just to let you know, well, that'll be a bit greenish at the start, and then I will come back afterwards. So this is one of these complex habitats. In the center, there's this reduced sediments, and you see all these clams. All these guys are clams that are living there. You see the size of it. The size of this iron circle is like 30 centimeters. So it's the size of a few meters in size, and it's all living clams that are working there with their symbionts. I will go into the details how they work afterwards. But you see that these are complex habitats over there. They're all living. You cannot see them moving, but they're all living. Yeah, there was one that was expelling some of its cloud of sediment. Okay, Paul, if you can go to the other one. Okay, this is another type of habitats over there. This is the reduced sediment. You see over around the bare sediment is everywhere, and in the middle you find this reduced sediment, very black sediment. This one, what we call it the giant head, so because of the nose here. And what you can see also around is that you have a lot of these stains of orange stains, probably linked to iron reoxidation, and also some whitish sediment here, whitish ecosystem, probably bacterial mats that are growing. A few juveniles can be seen in there, not very well seen, but they are inside. And so we believe that this is probably the start of the colonization of the by the symbiotic bivalve in this area. So yeah, next one. So the next one is about another type of habitat, which is made of more complex, made of crust, iron crust here, black sediment, so reduced sediment, and also whitish stains of bacterial mats. And the fourth one will show you what the bare sediment look like. So the usual, okay. Okay, this was Dalcom. Okay, this was most of the surveys we did was on the bare sediment. And you can see here and there some of these orange stains almost everywhere. There's probably linked to burrows that expand some of the iron that is oxidized over there, and some fauna here, fish and shrimp. Okay, so this is okay. My turn again. We're here. All right. So thank you, Paul. And just to okay. Just to tell you a little bit more about this chemosynthetic fauna. So we had a number of papers that were published in this deep sea research special issue. It's made of chemosynthetic bivalves. They live in symbiotic with the symbiotic bacteria that get their energy from the oxidation of H2S and O2. So they need the H2S to live and thrive. And the availability of H2S determines the species settlement, the succession traits. If they have the individuals are large or small, the affinity of the hemoglobins for O2 and so on. So there were a lot of studies that were done on that, and we could come up with a proposition of succession of habitats going from the reduced sediment that I showed you before to the microbial mat, and then the vesicomid settlement and size over the different size. And this goes with the H2S maximum depth. Okay, I will accelerate a little bit. It's already half an hour. So we also investigated the biogeochemistry of the sediments, and what we found was an extreme heterogeneity, which was really related to these habitats. If you look at the bare sediment, it's mostly dominated by oxic and suboxic diagenesis, so oxygen and iron in lava sediments. If you look at here, oxygen decreasing over 1 centimeter, so that's a pretty large consumption of oxygen, and a lot of iron. I told you about these orange stains in the sediment, a lot of iron in the pore water, up to 600, 700 micromolar of iron here. You see that there is some recycling of organic matter with the DIC increasing up to 8 millimolar, so showing that that organic matter is decomposed in the sediment, together with increase in the phosphate and ammonium. So that's for the lobe entrance at site A, and it's very similar on all the levees in the sediment. this extreme heterogeneity, and oh, one thing I forgot, there is no sulfide here. So sulfide is these triangles, and you cannot see triangles because they're all on the zero axis. If you go to the habitats where these bivalves live, then there is it's very different. To have a lot of sulfate reduction, large decrease of sulfate in the sediments, and a very large concentration of H2S, up to 2 millimolar, 22,000 micromolar. So there's a very large difference between these habitats and these levee bare sediment locations. Organic substrate does not show large heterogeneities between the different places, so that was a surprise. We looked for it and we didn't find really large changes between outside and inside these habitats. And so we at this day we're left with little explanation why we have these extreme gradients. So we know that there's some sort of a flank effects with some of the reduced material coming from the flanks of the channel. There could be also prior heterogeneity below the investigated sediment, but we did not have access to that prior heterogeneity. So it's still debated why and how these heterogeneities can arise in such an homogeneous environment. Okay, now if we come to the organic carbon budgeting, so as I said, we well, that's here. We will recalculate the input flux by the recycling plus the burial, and I will start with the burial. If we look at the organic carbon content in site A and site C, for example, so the entrance and the active lobe, we find that the organic carbon content is high, 3 to 4% in mass at all sites. So it's quite homogeneous between the sites. That the C2N molar ratio is above 10 for all sites, 10 to 15, sometime nearing 20. And the delta C13, where is it? This one is below minus 25. So minus 25 to minus 27. So especially at the site C. I forgot to tell that most of the sediment is a fine grain sediment. With average grain size below 10 micrometer. So we have a quite homogeneous medium in the levee, in the bare sediments, and with a large concentration of organic carbon. So if we look at sedimentation rates, I will start with the right panel here. With the site A at the entrance on the levees, we found that the sedimentation rate was between 0.6 to 1 centimeter per year. So think about it, that these are already very high sedimentation rates, probably 100 to 1,000 times more than the average abyssal plain sedimentation rates. But if you go to the last the active zone, the presently active zone, then you find that, so this is over 6 meters. We find excess lead to 10 for those who know or are familiar with these concepts, and also cesium-137. So that means that over 6 meters, we have like 60 years since cesium-137 has come with the bomb test in the atmosphere, and this was depicted in the 60s. So it's a 60 years and 6 meters of sediment deposited. So this is really huge. And this gives rise to sedimentation rate 10 to 20 centimeter per year. So this is like top set of a delta, yeah? But that's 5,000 meters below the sea level and 1,000 kilometers away from the shore. So if we calculate now the burial, it's really extreme. So I did the calculation here in a paper that we wrote with all this bench of co-author for quaternary science review, where we put together all these mass balances, and we were able to calculate the organic carbon burial, an average of almost 140 gram of carbon per square meter per year. So if you compare that with the actual burial rates in the Atlantic, South Atlantic Ocean, it's 1,000 to 10,000 times larger than in the surrounding abyssal plain below 3,000 meters. So it's really a huge, huge burial center. And this is mostly linked to the fact that compared to the hemipelagic trend of sedimentation rate here decreasing offshore, which is normal because all the sediments are deposited on the shelf, the slope, and they don't go much further in the abyssal plain. Over there in the lobe and the canyon and the lobe zone, you have an increase of sedimentation rate. The furthest you go from the river mouth, the highest the sedimentation rate, and it goes. This is a logarithmic scale, so from 100 to 1,000 to 10,000 centimeter per kilo year, and so this is why this is such a large deposition of organic carbon, burial of organic carbon in these sediments. Now, if we go on to look at the recycling, I said before that it was based on oxygen demand of sediment multiplied by the C to O2 ratio. We took the C to O2 ratio of 0.9 because the C to N ratio is pretty low in these sediments, and so the 0.9 seemed appropriate for this setting, and we recalculated the oxymineralization rate. And what you can see that compared to well, if we compare these total oxygen uptake rates to the rest of the South Atlantic or the South Atlantic nearby, it's 5 to 10 times larger. So this gives credit to the pretty large biogeochemical dynamics of these sediments. But despite the fact that the oxygen uptake rates and the carbon mineralization is larger than in other places of the South Atlantic, the recycling is still much lower than burial. If you look at the number here, 26 versus 140, so it's 5 times lower than burial. But for those who are familiar with the burial and recycling in abyssal sediments, it's generally the contrary. Generally, there is 10 times more recycling than burial in abyssal plain sediments. So here we have the reverse, much more burial than recycling. So this again is linked to these very high sedimentation rates that we find there and the huge accumulation of sediment. I told you about the reduced sediments and the ecosystems that live in there. They account only for a small part of the total recycling because they occupy only a small surface area. But the oxygen uptake is 5 to 20 times higher, of course, because all these reduced sediments and these animals that live in that particular spaces. So if we now sum up all that these numbers for all the sites, so we had like 5 sites and the average over there, so we can come up with a total of for the organic carbon. So that's the total would be the deposition of organic carbon from both the vertical and the lateral flux. So and we can compare this number, the total deposition of organic carbon, to the vertical flux that was measured by the sediment traps. And this measurement gave us only 1 gram per square meter per year. So it's only 1% or less than 1% of the carbon that originates from the vertical flux captured in the sediment traps, meaning that a large majority, probably 99% of the flux is coming with the turbidity current and probably of terrestrial origin. I will come to that point later. So if you combine these numbers with a map that shows the spatial extent of the different lobes, so like site A was attributed to lobe 1 and 2, then site B like lobe 3, etc., then you can calculate the overall burial and recycling, so the overall input to the lobe. And that's what we did in this this paper in Quaternary Sound Review, and we calculated that 0.42 teragrams per year of carbon are exported and deposited in the lobe region every year. So a part of that is recycled, like it's less than 20%, and 80% of that is buried in the sediment. And very quickly, I will come back to that later, but you can compare the burial here, 0.35, which is only one part of the burial, but the burial just in this lobe region to the whole South Atlantic burial, and it's already like 20%, 25%. And if you think about the surface area of these terminal lobes, it's only 0.01% of the South Atlantic. So it's a very small area, but buries a very large flux of organic carbon. One thing that is also interesting here is to know what is the origin of this carbon. And so for that, we looked at several markers using palinofacius, using delta C13. Here I will only show delta C13, looking also some biomarkers of the sediment. I will only just show you a brief part on the delta C13. You see here this brown part here is the terrestrial organic carbon, so minus 26, minus 26.5 here, and you see that along the path of the canyon is paved of this terrestrial organic carbon in the sediment, and especially in that region, which is the lobe region, the lower lobe channel system and the lobe system. So, and interestingly, in that region, the further you go away from the shore, the lower the delta C13, which indicates that more and more terrestrial organic carbon is deposited in these sediments along the canyon axis and down to the lobe zone. But when you go away from the channel and the canyon and the channel lobe system, then you find a normal sedimentation with lower and lower proportion of terrestrial organic carbon on marine organic carbon everywhere. So in that region, the marine organic carbon contributes only a small percentage. I showed you that maybe 1%, as expected from the fluxes, maybe a bit more than 1%. There might be some marine organic carbon entrained with the rest of the turbidity current. This is still debated, but at least 90% of the buried carbon is of terrestrial origin in this area. Now we'll come to the final calculation of the source to sink system for the Congo system. So we have the lobe burial. I told already 0.35 teragrams per year of carbon, and we have also the upper and lower levees where quite some canyon sediment and organic carbon can be deposited and buried. So the upper levees do not contribute much because of their small sedimentation rate, but the lower levees contribute almost as much as the lobe burial, which gives us a total of 0.74 teragrams of carbon per year. And if you compare that to the river discharge, which is calculated to be 1.9 teragram per year, it's almost 40% of the river input that is buried in this sediment. So it's really a lot of sediment that is a lot of organic carbon, terrestrial organic carbon that is buried in this complex, not only in the lobe complex, but also in the lower levee. And that needs to be taken into account in organic carbon budgets. Okay, I will go now to my conclusions. It's 15:45, very good. So the terminal lobes of the Congo Canyon, I find they're really a fascinating environment. They host a number of habitats based on reduced substances, like microbial mats, chemoautotrophic bivalves, which were unknown in such environments before. And these habitats, they thrive in anoxic environments, which is not very common in the deep sea. They are very active, biogeochemically speaking, in the deep ocean. And some of our colleagues thought that these reduced environments in the middle of the abyssal plain could probably serve as stepping stones for species migration, species that need the anoxic environments to migrate, especially these chemosynthetic bivalves. And they also, of course, represent, I showed that before, a mega burial site of terrestrial organic carbon, which was ignored before this study. And it represents 40% of the Congo River input, and probably 1/3 of the total burial of organic carbon in the South Atlantic. So there are still unanswered questions on this topic, and like the spatial distribution of the lobe habitats is still very poorly constrained. We think that the flanks seems to play a major role, but it's not very clear, and we don't know how the generation of this biogeochemical heterogeneity can happen in an otherwise pretty homogeneous environment. So we thought that there could be previous hidden heterogeneities behind the habitats, which we didn't find arguments for that or evidence for that, but there might be previous heterogeneities. There might be also bioengineering species. So probably these bivalves, they play a role in maintaining the anoxic diagenesis and keeping their sulfide concentration for the bacteria. And also, I think one of the most unexplained thing is the effect of the pulse events and the temporal dynamics of these systems. So we know that more generally that in these source to sink, these delta systems, there is a lot of temporal variation, and they are very often, except for the very human regulated rivers, but they are very often dominated by extreme event or intense event like flood deposition. And so the pulse events are really important in driving the biogeochemical, the sedimentological behavior of these environments. And so, like Paul Tolling told about the massive deposit that happened in 2020, beginning, I think it was in January and March, I think, and there was a massive turbidity current that deposit a lot of sediment in this, probably in this slope zone. So we would be very glad to go back and look at the environments we found there, how they survive, if they survive, and how the recolonization is going on. So that would be very interesting to see. We have also here in my group, we are also working on this effect of pulse event on the biogeochemistry of sediment using models to try and see how the sediment and the biogeochemistry can adapt, react to these massive events, to these pulse events, both in the deep sea and also the deltaic systems. Okay, well, I think I'm down to the acknowledgement. I think this was really a great project, thanks to this Congo group that we see here on board the ship at the end of the cruise. I think they raised their hand because they're so happy that the cruise is finished. And but no, I think that was really a great cruise, a great project that we had. There was a multidisciplinary approach. This is also something that is really needed in these systems, both geology, biology, microbiology, organic geochemistry, inorganic geochemistry. So it was really a huge program, and multi-PI with some American colleagues. I've seen Martial Taillefer here attending the talk, and also all the French colleagues over there, funding by the ANR in France to the Congo project for 5 years. So that was great. So thank you very much. And now I welcome your questions. Thank you.
Thank you, Christophe. That is fantastic. I will have to say your team did a great job, because in this Sync talk series, we have a couple talks talk about the canyon and material transport, like Peter Torning's talk, and Charlie Paul and Peter Cliff talk about Indus River, and even Sediment talk a little bit Gulf of Lyon, the canyon transport, and also in 2 weeks James Liu will talk about Taiwan, the Gulfin River. I think it is great. I know that a couple of people have to leave in 10 minutes. If you have to leave, I know Jim Best and any other. If you have a question, please don't feel shy. Jump in to ask. And Christopher, try to keep it shorter so a couple more people can ask questions before 10 a.m. East Coast. So, and I see Tom, Bob Ella, and if you two can stay after 10, then you can maybe wait for those who have to leave first. But if you also have to leave, go ahead to ask. Anybody?
Oh, I do have to leave. This is Bob.
Okay, Bob, you okay?
Christophe, that was great. And one of the things, I have a couple questions, and I think they're both pretty brief for you to answer. One is, do you have any DIC measurements of pore water Del C13?
We have some measurements of of delta. We did also delta C14. Unfortunately, the data were pretty messy, so I don't know exactly what to do with them. But I would be very glad to share that with you for your insight. But we have that.
Okay, I was just interested.
It's not very clear. Yeah.
Okay. I was interested predominantly in whether there's a lot more marine remineralization taking place than you might think from a solid phase. The other question I had concerned the depositional dynamics from your site C. And you talked about accumulation rate of like 20, 30 centimeters a year, something like that, or in that general region. And how do you know that site looked like it might have just been 8 meters almost at once? I mean, how do you, there wasn't much gradient in the isotopic values of the lead-210 or cesium.
Yeah, well, we don't know, but it could well be that because, like, these lobes, they are prograding. So you see the first one is on the east, and then it progresses to the west. So it could be that that maybe like 50 years ago, there was a massive, giant turbidity current that filled this of a cuvette over there. So it could probably be filled with that amount of sediment, and then now it's progressing to the west. So to the so it could be, it could be.
I remember on the Amazon Delta that we had a couple of sites like that too that seemed to be accumulating multiple meters in single events, or basically single events. So very, very interesting. Thank you.
Okay, great. Who is next?
I sort of have a similar question. So, these bivalve photo looks really impressive, but Christophe, when you're doing pore water sample or in situ measurement, in particular microelectrodes, you have to be away from that, right? And so how do you really integrate the result or interpret, put this spatial heterogeneity issue into consideration?
If you look at these, I just showing again the one of the feature. This one we call the mouth. So because it looks like lips, well, whatever. So so that's the poetic part of the talk. So what we did, we tried to make so in these very reduced environments, the bivalves are most of the time around it. I suppose they can be also poisoned by too much sulfide, can poison them. So they keep near the sulfide, but not too into the sulfide, so they don't get poisoned. So what we did, we made measurements, for example, here, in situ measurements with the sulfide, and also we took cores also in that area where we could make micro profiles of oxygen and sulfide. So, but of course, in the you're right, in the bivalves themselves, you cannot make micro profiles, because then you would break everything. So yeah, you're right.
Okay, good. Tom.
Yeah, fantastic talk, Christophe. Really, really interesting. So I agree with Bob that those radionuclide profiles did look pretty steady, or didn't have a pulsing to it, which made me think the same thing in terms of a big event, but just a big depositional event. But if you are arguing that these sedimentation rates are that high over more constant levels, rather than this periodic pulsing, how do the bivalves in that community sort of keep up with some of those rates? I realize that they may not be in the real hot spot for that, but it's still high in there. Are there any?
Yeah. Actually, they are on these sediments that have like 10 to 20 centimeters per year. There are some. So this mouth that I showed before, we have also another one which looks like a heart, which is also in that particular place where the sedimentation, well, the average sedimentation rate is that high. So it's hard. I agree that it's hard to understand how they can keep up with with such a sedimentation rate. If you have like 20 centimeters of sediment that pile up on their head, like every now and then, and sometimes, so so this argues for maybe a sudden emplacement of sediment and then colonization of by the bivalves. Actually, what we did when we did measurements was, so we had two crews, one in February, one in December. And so the first one, we did some measurements, and we disturbed some of these environments. Although they are so spectacular, we didn't want to, but for measurement's sake, we had to. So we disturbed, and when we left, there was such a cloud of sediment above that, we didn't know how we left it, but probably not untouched, if you see what I mean. But when we came back the year after, it has recovered very nicely. And so, so I suspect, well, you can see them spitting the sediment after, if some is deposited. So I suspect they can, as long as they have sulfide, they can live with perturbation, all the animals. Probably not if they have 1 meter of sediment above their head, but so I don't know. And for the deposition, it can be that it has been emplaced in 1 or 2 events, So I would be glad to go back and look at the last event that Pete Tolling told about in 2020, because that would be very great to see where it has gone and how far, and if we still see. We put some markers, I showed on one of the pictures, some markers. So if we still see these markers, and so yeah. So we need to organize a cruise, but that's a long work.
Yeah, that's fascinating. This really thought-provoking.
Okay, cool. Christophe, you show here the sedimentation rate is 12 to 22 centimeters per year. That's really, really high, because most of the world, the delta offshore is up to 10 centimeters. Here, the really is kind of is end member, almost, but also you show, the lobe. The lobe show the terrestrial carbon, and also show the, the marine carbon kind of sharply decreased compared to the terrestrial carbon. Terrestrial carbon can transfer much further. So did you think? Did you compare with other river system for example, the Ganges permaculture. You know, I scheduled a Valley to give a talk about Ganges, but maybe we have to reschedule him. And other system is how about the nutrient from Congo compare other reef system? Is the nutrient lower? Why the marine carbon, you know, decrease so sharply? Is the primary production limited, constrained? This is a question.
Yeah, I'm not really sure about that. I think compared to what I read some of Vallier Galli's work on the Ganges Brahmaputra system and the Kenyan, the deep sea work they did. I understood that the system in the Ganges, this system, this Congo system, is really for I don't know for what reason, but it's really channeled to this very small area. It's only 3,000 square kilometers, the lobe area. So it's really, really small. Yeah.
Yeah.
And especially if you think that there's only one part of this 3,000 square kilometer that is really active. All the rest gets a little bit if the flow is large, so it's spinning over. But otherwise, everything is channeled to this last lobe. I understood from the Ganges Brahmaputra that the system, once it goes to the fan, is more dilute. So it spreads over a wider area. So of course, then the predominance, the larger concentration of organic carbon can be lost with the marine primary production. I'm not sure that it's a question of the primary production, because over there in the Congo has also a large primary production plume and nutrients. So
you know, another thing you need to take a look is the tide, because based on my understanding, the Congo River is only about macro tide, only 1 meter tidal range on average. So the tidal range is so small. But compare Amazon, you know, compare the Ganges, even the Irrawaddy in Andaman Sea, and Yangtze, you know, Yangtze East China Sea, there's a big tidal range. When they have big tidal range, you know, the interaction of the seawater and the fresh water, they just back and forth. So the nutrient utilization may be much higher, you know, I don't know, maybe we need to put them together, do a comparison.
Yeah, that could be interesting. I will look again into the work on the Ganges Brahmaputra deep sea fan and try to, you know, make a short comparison. Then we can rediscuss that.
Yeah, wonderful. You know, after this talk series, we will have maybe 10 talks related to the Congo, to the Kenyan system. Maybe we can put a special issue, maybe write a review paper. You know, I think that would be a good collection.
That would be good.
Okay, is there any other question from audience? And if not, I think I will have to stop a little bit earlier today, because you know the moving company, the waiting moving my table, my computer, try to disconnect my internet. I said I'll put them down. So it's great. Once again, this talk will be online on the YouTube, and if you or your student want to rewatch, definitely many people will rewatch your talk. That's a great work. And also, as I mentioned in the beginning, if anybody, your student, postdoc colleague, I even appreciate if you can volunteer to give a talk. That would be great. And even if you can recommended some senior faculty or retired professor, you know, I want to occur their best of the best of their career. So, you know, because they maybe never have a talk presentation on the YouTube. So we can make it happen. Okay, great. So thank you very much. And I think we'll end it here. Hugh, if you appreciate, if you can stay here a couple of minutes, then we can, you know, test the Zoom and make sure everything okay. Christophe, thank you. Okay.
Thank you very much.
Hi, Paul.
Hello, Hugh. That's great. Let's see. I can make you. Or you can try to share screen and.